pretty clear that genetic drift is actually the main factor here that's driving the patterns of population structure that we see. But what that means is that also where we have isolated populations and the environment differs, that we also should expect to see some strong pattern signals of local selection. And across the area where the samples are, the main sort of climatic gradient is basically a gradient of aridity. So in the east, so in the east it's sort of more fertile and a lot wetter and a little cooler, and then gradually as you move further west it gets drier and more arid until you get right down to the mouth of the Murray near Adelaide, which is where we're from, and it's really pretty dry down there. And there's other things like differences in the flow regime across there as well, and other human disturbance like land use that probably impacts as well. So, as I said, this is part of my PhD project. I'm going to be talking mainly about landscape genomics today. Just quickly though, a couple of other aspects. The main chapters of my thesis are to do with sort of global gene expression in both wild and captive populations of these fish, and also the genomic consequences of captive breeding. So, at Flinders Uni, we manage a conservation breeding colony of these and another sister species as well. And so I've got several generations of those, plus a reintroduced population that I'm kind of exploring as well. But today we're just talking about landscape genomics, and I've only really had my whole data set for a few weeks, so these are pretty preliminary analyses here. But in landscape genomics, we're really asking the broad question about how the spatial patterns of both neutral and adaptive genetic diversity are associated with patterns of environmental variation. And so the couple of things I'll talk about today. One, we're trying to answer the question about how modifications to the flow regime might be impacting natural evolutionary processes like connectivity and gene flow, and also then thinking about which environmental factors might be important in local selection. So I prepared TD-RAD libraries for 464 individuals, 100 base pair and Illumina sequencing and multiplexing 48 samples per lane. I used a relatively new bioinformatic pipeline, Descent, to process the raw data. Now the really cool thing about this is that it includes a proper alignment step, which is a little bit different from stacks, which we've been using previously in the lab, and that really helped to solve some of the issues I was having with missing data amongst my really divergent populations. So I'm happy to talk to people afterwards about that. Basically, we ended up with a catalogue of 2.5 million SNPs, and the data I'll be showing today were all biallelic SNPs, 1 SNP per locus, and filtered for 70% of individuals in 70% of the population. So we end up with a little over 5,000. So that's just the sampling we have across the distribution. That represents pretty much all of the known populations in the Murray-Darling, and they're just grouped by colour there in the catchments that the sampling locations occur. I should say there's 26 of those. So we jump straight to the results here, and we look at some population structure analysis, just basic stuff. So DAPC and fast structure are both in pretty good general agreement about what a population is here. And the main thing is that there's a really distinct populations, no matter which method you use, and that these populations align really tightly with the individual catchment boundaries across the basin. And we see this pattern repeated over and over again with other freshwater fish and other animals as well, both with other work in our lab and other people's work as well. And so when we want to move away from population genetics and move into landscape genetics, or genomics in this case, we're really asking the question about what features of the landscape might be influencing the patterns of population structure that we actually see. And a really good model to explore this, in my case, is stream tree. So basically you give it a model of your stream network topology and a pairwise FST matrix, and it tries to estimate the contribution that each stream segment makes to FST. And so when we look at the observed pairwise FST and the model fitted FST there, we can see it's a really good fit for our data. And then when we map it onto the stream network, what we can see here is the yellow sections of stream are those that the model says are contributing the least to FST. through the orange and then into the red sections, which the model's suggesting in some way are influencing FST to a greater degree. And this is a really useful kind of tool for exploring your data, and when we think about what might be making these patterns, we can add the larger reservoirs in the system, and we find that in every case there, wherever there's a higher segment that's associated with higher FST, there's also a large dam or reservoir. And then, even if we want to go a little bit further and start thinking about a smaller spatial scale, so within catchments, we can go back to the data set that I showed at the start and maybe add in the smaller farm dams as well. And I haven't gone as far yet as actually doing any empirical testing here, but there's some really kind of interesting patterns there that I'm sort of, the next step is to go and test them a little further. So the next thing I want to talk about is how the environment might be influencing local selection. And the most common method for doing this at the moment are the FST-based outlier tests. But when we have a global FST approaching 50%, they tend to give you a lot of false positives, and you end up with the results that look like that. So in the case of loss 10, we apparently have more outliers than we do neutral loci, and it doesn't get a whole lot better when we use algorithm, which can supposedly cope with some degree of hierarchical population structure. So the way that we've gone is to start using a genotype by environment approach. And what we're doing here is looking for direct correlations between allele frequencies and some environmental variable of interest. And so the method we've taken is a fairly new one called GenLand, and the great thing about that is that it can account for the shared population history and the hierarchical structure we find in this system because it's spatially explicit. So essentially, for each locus, it does a Bayesian model test of two competing models, one with the influence of the environmental variable and one without, and both of them with a random spatial effect. And so if we think back to our gradient of aridity here, so fertile in the east and more arid in the west, I've used net primary productivity as a kind of a good proxy for that here. If we run that through GenLand, then we end up with what I think is a fairly conservative threshold here with using everything above a log base factor of 10. We're considering has strong support for the model with the environmental variable. And if we just think about one of those markers and we map the allele frequencies back onto our environmental layer, what we find here is that the blue allele seems to be favoured more in the more fertile areas here, and then it changes as we get drier into the yellow allele and then gradually moves over to fixation in the driest areas in the west and also in the north, which is really dry as well. So this approach is kind of just the beginning, and the next step, as I said, we're doing some RNA-seq as well, and we also have access to a transcriptome from another local fish from the same family. And so once we sort of get together a list of candidate loci, then we want to start going to the next step and trying to work out what are the genes doing that these markers might be linked to, and try and work out actually some kind of adaptive, what's going on in the adaptive landscape, I guess. So just to sum up, so basically I'm using a landscape genomics framework to try and identify environmental factors that are contributing to the spatial distribution of genetic diversity. I think, as I said, this is all really preliminary stuff, but we do seem to have some support for the idea that human modifications to the flow regime are probably disrupting evolutionary processes such as connectivity and gene flow. And once you get that, you start getting an increased risk of local extinctions in demes, and then it's really unlikely that they're going to be recolonised from neighbouring populations, and then you start to lose the species regionally. And we're already seeing that there's a number of these sampling sites that there's no picking perch there anymore. And the other thing is we have evidence for local selection within populations across a climatic gradient. This has implications in the future, thinking about climate change. The arid region is expected to keep encroaching further to the east here. And thinking about which populations might be resilient to that change, and even thinking about conservation management. They're already considering translocations among some of those populations, and maybe having an idea about which source populations might be best is something that we're looking into as well. So just some acknowledgements there. So Jonathan, Kat and Renami in the lab, and Luciano and Luciana, my supervisors, and especially the ARC and Flinders University for some funding. Hopefully we'll have time for a couple of questions.